Hi, I am Dr. N. PACKIALAKSHMI, Assistant Professor, PG and Research Department of Microbiology, Jamal Muhammad College. Now we are going to learn the basic techniques for our microbiology laboratory. Let's go for a practical session. First one, fumigation. Fumigation is a process of gaseous sterilization which is used for killing microorganisms and preventions of microbial growth in air, surface of wall or floor. Procedure: take potassium permanganate 15 gram in a petri dish, add 25 ml of formalin solution to potassium permanganate, immediately fumes or comes. It is toxic to inhalation and ingestions and causes irritations to eyes and respiratory tract. So immediately close the area. Keep the room under fumigations for a minimum of 8 to 12 hours. Now the lab ready for our microbiological work. Nutrient agar medium preparation. Nutrient agar is a general purpose nutrient medium used for the cultivations of microbes. Procedure: take 100 ml of distilled water in a conical flask. Add 0.5% peptone, 0.3% yeast extract, 1.5% agar, 0.5% NaCl. After mixing the ingredients, to check the pH. pH is very important for microbial growth. Now, the pH is adjusted to neutral with the help of pH meter. This is buffer tablet pH 4 and 7. pH meter. pH meter is an electronic device used for measuring the pH of any solution. pH meter calibration is required at regular intervals. So, first calibrating the pH meter. And for calibration purpose, we are taking two color-coded standard buffer solutions of pH 4 and 7 for calibration. Take 100 ml of distilled water, add 1 buffer tablet of pH 4 and mixed well. Now the pH 4 buffer solution is ready for calibration. Next flask, add pH 7 buffer tablet. Mix properly. Now the pH 7 buffer solution is ready. Now go for calibration. Dip the electrode in the pH 4 buffer solution. Rotate the function knob to check the pH mode. Adjust the pH using calibration knob and finally go for checking mode. The checking mode display shows that 0. After finishing the process, without using calibration knob to check the pH. Now the electrode removed from the buffer solution. Wash the electrode with distilled water. Dry the electrode with tissue paper gently. Dip the electrode in the another buffer solutions of pH 7. Rotate the function knob to check the pH mode. Adjust the pH using calibration knob and finally go for checking mode. The checking mode display shows that 0. Remove the electrode from the buffer solution. Wash the electrode with distilled water and dry the electrode with tissue paper gently. Now check the sample. Press the pH mode. Wait for some seconds until the reading stabilized. In this sample, the pH is 5.8. We need neutral pH, so add 2 or 3 drops of alkali solution to adjust the pH 7. After checking the pH, plug the cotton plug in the conical flask. Next, using micro oven 20 seconds for proper dissolving the nutrient agar medium. After finishing the process, proper wrapping for conical flask and petri plates. Now the nutrient medium and petri plates are ready for sterilization. Autoclave. Place the nutrient agar medium and petri plates in the autoclave. Close the lid. Tight the screws properly. Switch on the autoclave. Autoclave. Use saturated steam under pressure of approximately 15 pounds per square inch to achieve a chamber temperature of 121 degrees Celsius for 10 to 15 minutes. LBS means l-libra balance scale. After finishing the process, loose the screws. Take the medium and petri plates for working. Laminar airflow chamber. Wipe the chamber using 70% ethanol. Switch on the UV light for 10 minutes. Switch off the UV light. Next, go for pouring. Pour 16 to 20 ml of melted cooled nutrient medium in the petri plates. After pouring, allow the medium to solidify. After solidification, keep the plates in an inverted position. Next, go for streaking. Streaking is a technique used to isolate a pure strain. The first streaking, quadrant streaking, label all the plates on the bottom of the agar plates using marker. Sterilize the loop. Withdraw 1 loopful of inoculum in the broth. Take the agar plate. Place the inoculum on the agar surface and streak the inoculum from side to side in parallel lines across the surface of area. Reflame and cool the loop and turn the petri plates to 90 degree. Touch the loop to a corner of the culture in area 1 and streak the inoculum across the agar in area 2. The same procedure follow on 4 corners. The next one, zigzag streaking. This is simple streaking. Take 1 loopful of inoculum in the broth. Place the inoculum on the top of the agar surface. Just streak 2 or 3 times and continue to track the loop in a zigzag formation. Next one, T streaking. T shape is drawn on the bottom surface of the plate using a marker. Take 1 loopful of inoculum in the broth. Place the inoculum on the top of the agar surface. Track the loop with suspension in a zigzag pattern in the top half of the T. Flame the inoculating loop once again. Rotate the plate at 90 degree. Track the loop lightly from the first section towards the second section and repeat the zigzag pattern. After finishing the process, next go for incubator. Incubate all the plates at 37 degrees Celsius in an inverted position for 24 to 48 hours. After incubation, the isolated colonies was absorbed in the plates. Microorganisms are everywhere, and to study specific organisms, it's essential to grow them in a strictly controlled lab environment. A complete sterile condition protects the pure microbial culture from contamination by other organisms entering through the air, water, or other unsterile sources. Thank you for watching this video. Thank you.